Topic of metagenomics, and the first speaker in this little mini session on metagenomics is Jonathan Eisen, and the title of his talk is, as you can see,
is too long.
Too long. It's a long title.
Fortunately, I have shortened it, and shortened it again. So really what I'm going to do is talk mostly about phylogeny in sort of the general sense, and how I think it should be used, or is being used, or in particular what we're doing in my lab on using phylogeny in analyzing some genomic data, but mostly data from what's called metagenomics, which I'll talk about a little bit more, but is where people go and collect samples from the environment, grind them up, extract the DNA, and randomly sequence those DNA samples from the environment, say soil or air or water, etc. And I know people here sort of know what phylogeny is, but I just want to make the point that when I talk about phylogeny, I don't mean necessarily a single bifurcating phylogenetic tree. So I mean that when I talk about phylogeny-driven approaches, and we frequently use sort of single bifurcating tree models for most of our work, what I mean, though, is the benefit of using the history behind the data. So all of this data that we're collecting from genomes and from metagenomes, et cetera, is biological data with a history embedded in it, and you've already heard lots of examples, and everybody talks about this all the time, but what I want to sort of reiterate is different ways you can take advantage of that history to learn more about the data than maybe other more direct approaches to analyzing the data is. And it doesn't matter, this is for Jim, here I took a figure from one of his papers on different models for rooting the tree of life. It doesn't matter to me what the tree is. I mean, there's going to be a constant flux in the exact phylogenetic model for a group of organisms or for all organisms on the planet. Again, what I want to emphasize is that trying to incorporate phylogeny and that history into analyzing this type of data is critical. And, you know, the more accurate your phylogenetic representation is, probably the better you will do, although you can introduce complexities if you try and make your models be perfectly reflecting the history. You might actually set yourself down some complicated paths for the computer science that may not always work out.
What is the alternative instead of using the phylogeny?
Clustering by similarity, for example, is one classic alternative. And that's usually what I am comparing to, is taking biological data. Everybody does comparative biology. We quantify the similarities and differences between genomes or between taxa or between genes. And what adding the phylogeny to that is trying to figure out the history of how those similarities and differences came into being, as opposed to just grouping things by their similarity. So that's the main contrast, although there are other approaches to dealing with the data where you can have a sort of non-cluster approach, historical approach, but those are sort of things I don't even understand in mathematical space completely. So one reason to do the historical-driven approach, phylogeny, as opposed to grouping things by similarity, is that rates of evolution vary between taxa, or between genes, or between different parts of a genome. And so if you had a model of genes evolving like this, where after the separation of some lineages into two different branches, you had one lineage that split off, and then its evolutionary rate slowed down for whatever reason. Doesn't matter. And in some other lineages, maybe the evolutionary rate sped up. If you now take the data from those organisms and you group them by their similarity, the similarity will not group organisms according to their evolutionary history. So 5 and 3 might be the most similar organisms. Let's say this is a species tree here, in certain phenotypic space. Let's say, you know, crocodiles or something that has slowed down a certain part of their evolution. In some cases, that's useful to know. So you may just want to know the similarity among organisms or among traits if you're doing certain types of experiments. But in many other cases, what you want to know is actually the order of the branching to determine common ancestry of organisms or common ancestry of genes. And what I'm going to do is show you basically some examples of this. And this applies, yeah.
So you're saying that 4 and 6 have changed so much because their evolution has sped up that they're actually genetically much more different from 5 than 3 is from 5?
Yes.
Because 3 and 5 both sort of stop changing.
So let's imagine that this is virus evolution. And if you plot virus evolution on a tree, well, maybe this is not the perfect example, but the branch for HIV would be off the blackboard here. And because its rate of evolution generally has sped up. But that doesn't actually mean in all cases that you don't want to know where HIV sits relative to other organisms. So it may be that 2 other viruses in the virus tree, if you measure their similarity of their protease genes, are percent similarity-wise, they're more similar to each other. But there were events that happened on this branch. And those events may be useful in determining some property of the organisms that descended from that position. So, these are generally called synapomorphies, and when people talk about plant and animal evolution, it doesn't really matter what they're called. But those events that occurred in the branch leading up to the common ancestor of a group may be important. They're not always important, and the similarity actually in some cases is more useful, such as if you're interested just in phenotypes and there's been a lot of convergent evolution, that may be what you want to measure. But actually knowing that convergent evolution happened is a useful thing. Does that make sense? So, and again, this can apply to species evolution, to gene evolution, to genome evolution, to the evolution of anything. Doesn't really matter. So what I want to do is basically go through a few examples of doing this in the context of environmental microbiology, in sort of genomic-y in some sense, sometimes not in complete sense. Please just stop me anywhere along the way. I don't need to get even remotely close to the end here. So the first thing I want to talk about is something called phylotyping, which is a really big deal in environmental microbiology and now in some other areas as well. So basically the principle behind phylotyping started about 25 years ago when people started to generate sequence data from ribosomal RNA genes from organisms that they could not grow in the laboratory. And the reason for this was that there are many microbes in particular that we can't grow in the laboratory. The way that people were classifying organisms was by studying them in the lab and either looking at their phenotypes or sequencing genes from them. And so people went to environmental samples, Norm Pace in particular, and started to generate sequence data from environmental samples with ribosomal RNA genes. And then they would try and place those ribosomal RNA genes in a phylogenetic context compared to organisms that they had information about. And this is generally known as phylotyping. So you go to the environment, you generate ribosomal RNA sequence data, and then you build a phylogenetic tree, and you place your organism in the context of other organisms. So this is an example of this. This is from my, I think I might have a picture here. Oh, no, I cut out the picture. So this is from my undergraduate research project. When I was an undergraduate working with Colleen Cavanaugh at Harvard, she studied these really cool chemosynthetic symbionts that lived inside the cells of various invertebrate species, and you couldn't grow any of these in the laboratory. And so we went to these organisms, ground them up, ran PCR reactions to get out ribosomal RNA genes, and then sequenced them to figure out what they were. And then you build an evolutionary tree, and for example, you see that this group here is all chemosynthetic symbionts. That's what the sim means. One sort of clade or maybe a polyphyletic grouping. And what this was really useful for was to see what the closest free-living relatives were to this group. And then you could work on that organism in the laboratory, design experiments based upon that organism, and then go to the field samples and leverage what you learned from the organism that you could grow more carefully to study the organisms in the lab. This became a huge deal in environmental microbiology and still is. Now this project took me 1.5 years to sequence one 16S ribosomal RNA gene. And other people did this exact type of thing, and it led to some amazing discoveries about microbes on the planet. So for example, it showed that there were many lineages of microbes, phyla, let's call them, that there were no cultured representatives in the entire clade. So they were only known through these ribosomal RNA sequences in the environment. By building phylogenetic trees, these ones not filled in are lineages of archaea or bacteria in 2002 that there were no cultured representatives, entirely known through these ribosomal RNA analyses, basically. And so that's the way it sat for a long time. People would build evolutionary trees of these ribosomal RNA sequences, sometimes very painstakingly. There were nice databases that were built, nice software tools. There's one called ARB. There's a bunch of other tools that people use to do this. And then we got overloaded, as we thought at the time, with sequence data. This was when we had lots of automated. I was at Tiger for many years, Craig Venter's sequencing genomics institute, and we thought 10,000 sequences was overwhelming with ribosomal RNA data, and we tried to figure out how to deal with this massive influx of 10,000 sequences. Sad in comparison that now an Illumina machine can do a couple of billion in a couple of days. But people basically panicked and threw away the phylogenetic approaches to analyzing this data because it was just too big. You couldn't do alignments, you couldn't build phylogenetic trees, most people just said, we have too many sequences, we're just going to do blast searches of the sequences, which is a similarity search to a database, and that's how we're going to analyze all the sequences. So this was in about 2003. It seemed like a bad idea to a phylogeny-obsessed individual like me. And so we started to work on automated tools that would take thousands of sequences and put them all the way through phylogenetic classification, just like in the phylotyping analysis. And we built one tool to do this. And then as we were working on the tool, people started to do now random shotgun sequencing from environmental samples. So instead of PCR amplification, they would go to environments, grind it up, and just sequence everything from that environment. And went to these environments. So we had this tool, and we started to run it on PCR amplified data as well as metagenomic data. And the tool, we called it STAP for small subunit taxonomy alignment pipeline or something to that effect. And basically what we did was coarse and crude. We took the sequences, we did searches against a database to try and find some relatives to those sequences. We had a set of reference sequences from all across the tree of life. We took those reference sequences as well as a few additional ones that seemed to be slightly more closely related to our sequence than other ones, and built an automated evolutionary tree of those, found what the nearest neighbor was in the evolutionary tree, and then zoomed in on that clade, built another evolutionary tree of that clade, and used that to figure out what the sequences were. Yeah.
Did you know there are homologs, these sequences?
Yeah, so sorry, we started out doing this with just ribosomal RNA data.
Oh, okay.
So we did blast searches with the ribosomal RNAs to find homologs either in the metagenomic data or in the PCR data. And then we had this automated algorithm that would go through, and the output was a tree. And then we would scan through that tree, to try and get taxonomy classifications, et cetera. Now, when we did this, each sequence was analyzed separately. So we would basically stream through each sequence from an environmental sample as an individual and not compare them to each other. We weren't sure how to do that at the time. And then we realized this was not a good idea. And so what we wanted to do was, instead of streaming all the individual sequences compared to the database, we wanted to do that, but then combine them all together and compare them to each other, in addition to comparing them to the things in the databases. And we ran into a big problem with this, which came from metagenomic sequence data, which is if you randomly sequence data from an environmental sample, you might have sequences from your environment that correspond to your gene of interest, but they don't line up with the whole gene, that relative to things that are in databases. Some might be on one side, some might be on the other. And then when you compare them to each other, we have non-overlapping sequences for some of this. So there was work at the time that suggested you might be able to deal with non-overlapping sequences with supertree or supermatrix methods. Initially, we punted on that, and we said, let's just take a section where everything sort of overlaps with each other and only analyze the things that overlap at a reasonable amount with each other. So we did this when Craig Venter started sequencing things from the Sargasso Sea and then sailing around the world. We started to run these algorithms on that environmental data so we can build a tree of all the ribosomal RNA sequences that are coming from this environmental data, just like you could do from PCR data. And you can use it to classify organisms and learn various things about the organisms in that environmental sample. The great thing about this random sequence data, which was unique and still is unique, is that it gives you data from genes other than ribosomal RNA genes. So people for 20 years have relied on doing this phylotyping on ribosomal RNA genes because you needed to PCR amplify genes from an environmental sample. And if you didn't know what was in your environmental sample to start with, the best way to go after finding something was to use what are called universal PCR primers that can amplify, say, all the small subunit ribosomal RNA genes from bacteria at once. And then you can sequence them and figure out what bacteria were in the sample. So we were dependent everything on ribosomal RNA for analyzing most of these samples. But that ribosome RNA is not necessarily the best gene for all phylogenetic analyses of all organisms. And the great thing about metagenomic data is it gave us other genes to analyze for the first time from environmental samples. So we picked sets of genes that had been shown to be supposedly robust phylogenetic markers for a variety of reasons, and went through and built algorithms to do the same thing that I just showed you with ribosome RNA to do with these other genes. So REC-A, we picked RPOB, HSP70, and whatever else is on this, EFG and EF2. And the nice thing about this is one of the things that people want to do when they go to an environment, you don't just want to do phylotyping. What you want to do is count how many sequences come from a particular group and estimate the relative abundance of particular groups. If we do that with ribosome RNA, it doesn't work very well in some cases because the copy number of ribosome RNA genes varies enormously between taxa. And so if you estimate a 10-fold higher abundance of a particular organism in your sample, you don't know if that's due to 10x copy number of ribosome RNA genes or 10x of the cells. Many of these genes that we looked at were roughly single copy genes, and they allow a better estimate of the relative abundance of taxa than ribosomal RNA data. So again, we do this all with a phylogenetic approach. We build phylogenetic trees and look for where in the tree a particular sequence is, and then we try and assign that to a taxonomic group, and then we can count how many representatives there are of that taxonomic group from the data. Now, as an aside, I just thought I probably should mention this really weird thing we found, which we found initially 8 years ago now, and just published a paper on it about 4 months ago, which was when people looked through this environmental data, they usually were very focused for particular reasons. So they would look through ribosome RNA data and say, give me all the bacteria from that data. Let's look for where the bacterial-like sequences group relative to other bacteria. Where do the archaea-like sequences group relative to other archaea? Where do the eukaryotic sequences group relative to other eukaryotes? And we wondered, is there anything else out there? I mean, that is, are there sequences from these environmental samples that don't group within any of the three major branches? Jim would just say there aren't three major, but relative for the ribosomal RNA data, we were looking for whether or not something would group outside of the three major lineages where ribosomal RNA sequences grouped. So we tried to do this with ribosomal RNA. We scanned through this ocean sequence data for novel ribosomal RNA sequences. We built basically an automated algorithm to do this. And we found about 100 sequences that seemed to group in between the three major branches in the tree. And then we looked at all of them, and they were all crap. So the alignments in general for these sequences were bad. So yes, in a phylogenetic tree with something that's a bad alignment, it looks weird. That's not really a novel finding. But we didn't know whether or not there really were novel things, because the problem was automated alignments of ribosomal RNA sequence data at the time were really quite bad if your sequence was distantly related from something else, especially if it was a fragment sequence, because a lot of the alignment algorithms were based either upon having a close relative or based upon secondary structure. If you don't have the full-length sequence, you can't model the secondary structure completely. So we had a horrible time with these very strange ribosomal RNA-like sequences getting any alignment out of them. Yeah?
Could they have been chimeric to PCRs?
So this is analyzing random shotgun sequence data that doesn't have PCR. You can still get chimeras in some of the shotgun data, but it's less likely. So absolutely, they could have been, I mean, we couldn't even convince ourselves that any of the alignments were even good enough to find that, let alone that, but unlikely that there were chimeras in this data explaining that pattern. So we decided, why not look at other genes? Because again, for the first time, we could look at any gene, not just ribosomal RNA. So we went and looked at REC-A sequences. I worked on REC-A evolution as part of my PhD, so I'm obsessed with REC-A, so that's always my first pick to look at for things. We built phylogenetic trees of the REC-A sequences compared to known REC-A families. And it's much more complicated than the three-domain tree of life for ribosomal RNA, but we found many lineages in the REC-A data that were distantly related from any of the lineages present in bacteria, archaea, or eukaryotes. That is, completely novel lineages of REC-A-like sequences in the environmental data. we found the same thing for RNA polymerase, completely novel lineages of RNA polymerase sequences. Now, when we did this for, I must have cut out the slide. When we did this for RECA, this was done in 2003. And again, we didn't publish the paper until 2011 for a lot of reasons, yeah.
Yeah, do they come from the same place when you look at different genes, these new weird things?
Yeah, so
What was the question?
do they group in the same place, these different genes? So the problem is that most gene families don't have the same phylogenetic clades represented. So we were analyzing everything, which included virus data. So it turns out that most genes, even at the time, had homologs in some viruses. And now there are these crazy viruses that have been discovered, like Mimi virus, that have homologs of everything, basically. So, but they don't each have the same thing. So it's very hard to compare the tree of RPOB directly to the tree of RECA.
Yeah.
So what we did instead was we tried to find genes that were linked. So some of these had assemblies that could show up, or had some type of binning data that could tell us what was linked to these unusual RECAs. And in some cases, they told us likely what something was. So I must have cut out the slide, but one of these this one is a novel archaeal lineage that was not well characterized at the time, but is now better characterized, the Thaum archaeota or something like that. This one had a bunch of viral-like sequences and has now been shown to be closely related to these cyanobacterial phage. And then this one, GOS1 and GOS4, every sequence that was linked to any of these sequences also showed up as being completely phylogenetically novel. We have no idea what these are, still, to this day. My best guess is that they're from unusual viruses that are out there that have some very strange forms of some of these genes. But it's consistent with the existence of other major lineages of cellular organisms that have not been characterized. We don't know. Again, my guess is that these are from weird viruses that have just not been characterized. Yeah?
You said that the gene trees aren't identical. What's your hypothesis for why they're not?
So it's not that the gene trees aren't identical. We weren't even comparing the gene trees because the taxonomic representation for the different groups. I don't know. I mean, I've done comparisons for some of these gene families, and they are statistically significantly different even when you normalize for taxonomic representation.
Any hypothesis?
Most likely lateral gene transfer of some of these. I mean, there are other possibilities. But, I mean, we're looking at 4 billion years of evolution here, and long branch attraction could also complicate this enormously because there are a lot of these lineages are on very long branches. So I'm not confident in the exact phylogeny of each of these, but we actually threw out individual sequences or sequences that were only 2 in a group. We only analyzed the ones where there were many that appeared to be from one clade, many distinct representatives from a clade, but we still don't know what they are per se. But again, you'd never find anything like this if you weren't doing a phylogenetic approach. You'd just do a blast search and say, oh, these look like RECA, and you'd be done.
So what are the functions of RECA and RPO?
Sorry. RECA is the core of homologous recombination. It is found in bacteria, archaea, and eukaryotes. There's in all of those species, RECA homologs are involved in catalyzing the strand invasion that is part of homologous recombination. There are RECA-like proteins in some viruses, too, that appear to do the same thing. RPOB is RNA polymerase B. It's one of the subunits of RNA polymerase. So again, we're analyzing this metagenomic data and starting to slow down with the 100,000 sequences that we had to analyze. Again, I'm panicked over Illumina data now, but at the time, even 100,000 sequences was very hard, so we wanted to build automated methods to do these protein family analysis, just like we had done with the ribosomal RNA analysis. And we built this tool called Amphora, which actually probably works much better than the ribosomal RNA analysis because the protein sequences tend to be much more conserved in many ways, and they're conserved at the primary sequence level in a higher level than the ribosomal RNA data. So what we did was we built hidden Markov models of sequence alignments of representatives of a bunch of different gene families, and then we would use those hidden Markov models to align new sequences from either genomes or environmental samples and we actually built an automated method to trim out the poorly aligned regions of that sequence data, and then you can build a tree with any sequence that you have a good and hidden Markov model for. And so what we did was we would do this for complete genome sequences. We identified 31 genes that are found in broadly in bacteria. There were other groups that actually identified 31 genes too that are not completely overlapping with ours, but basically 31 housekeeping genes found throughout bacteria, some other genes found throughout archaea, and you can build a concatenated genome tree from this data. I realize that concatenated genome trees are not necessarily the optimal thing that you want to do, but this was very easy to automate at the time. And what we used this genome tree for mostly was then as our reference tree when we streamed single sequences from environmental samples against these hidden Markov models. We would lock a reference tree from this whole genome concatenated alignment and then ask for the phylogenetic position of each of the new sequences relative to that reference tree. Sort of similar to what we did with the ribosomal RNA data. And that allows us now to classify in a very high throughput manner sequences with phylogeny from metagenomic data and assign them to phylogenetic groups and use phylogenetic approaches to analyze the data. Now, this is pales in comparison to, is it Megan or Megan?
Stefan likes to pronounce Megan.
Megan. All right, so neither. Megan. So we were analyzing these sort of housekeeping genes, and there's this tool that they developed that's really nice, which can analyze all the data that you have in metagenomic data, and in essence do a similar conceptual activity, which is assign it to taxonomic groups and count how many sequences get assigned to taxonomic groups. It does a slightly non-phylogenetic algorithm, which is it's not building evolutionary trees of each individual sequence. It's looking at it's sort of doing the LBA, right? I mean, it's doing LCA, yeah, long branch attraction algorithm. That's what our method does, unfortunately. So it's a very nice tool for analyzing massive amounts of data. It's just slightly not as phylogenetic as sort of the Amphora tool that we were doing. It's much more high throughput. We did some comparisons. It seems that Amphora has got some slight performance improvements over Megan in terms of specificity, but not much else. But as a phylogeny-obsessed person, we're always trying to develop tools that will give us a phylogenetic tree of the sequences, not just because we want to use that for the taxonomy, but because we want to use the phylogenetic trees to ask questions about the tempo and mode of evolution in those gene families, in addition to assigning them to groups. And one area where people are doing that a lot is something called phylogenetic ecology. So this is sort of a standard thing in plant and animal world and now moving into the bacterial world. And I'm going to take you through some of the things that are done in that in the bacterial and archaeal world. But basically, the first step in what you could call phylogenetic ecology is trying to take the sequences from an environmental sample and assign them to clusters that could correspond to species, although not doing the same algorithms that you just heard about in terms of species. But a standard operating procedure in environmental microbiology has been to take sequences from ribosomal RNA in particular and group them by their similarity to each other and then get clusters from that and call them operational taxonomic units, or OTUs. So basically you're taking a bunch of data and you're finding sets of sequences that are close in similarity to each other and you're calling those units, OTUs. And that's very powerful, but what I think is more powerful is to try and analyze those in the context of the phylogenetic history of those sequences and not just identifying species. And this is what's being done extensively in the plant and animal world with this what's called phylogenetic ecology. So rather than comparing species lists between communities, lists of taxa that are present in communities, you want to compare the phylogenetic patterns between communities of organisms so that if two communities have nearly identical taxa that maybe are different species of a set of genera, that would tell you one thing versus if you have very distinct organisms that are in different phyla in a particular community. So another reason to do this is that that OTU clustering doesn't always work perfectly well, especially when there are variation in rates and patterns of evolution. So other algorithms can improve upon finding OTUs by going through the tree and trying to find, in essence, monophyletic groups within the tree that correspond to a particular depth in the tree, rather than clusters of things that have a set % similarity to each other. Finding actual clades sometimes tells you a slightly different story than these clustering algorithms. So we've been very interested in phylogenetic approaches to identifying these OTUs in this data, and we had, again, the same problem, which was we wanted to analyze massive amounts of data to find these OTUs and build phylogenetic trees, but the sequences didn't overlap with each other necessarily. And now, rather than throwing away some of them and just taking the remainder, we really wanted to know whether or not a sequence like this came from the same clade as maybe a sequence that only had a portion over here that didn't overlap at all with each other. So we basically have been exploring, although not completely using, methods like supertree and supermatrix methods to try and deal with this data and put them all on a single tree. Tom Sharpton, who's in Katie Pollard's lab at UCSF, and I helped with this, wrote a method to, sort of a jury-rigged method to do this with ribosomal RNA data that he called PhyloOTU, and it basically doesn't do a complete phylogeny of all the sequences. It actually takes a little shortcut in using something like this method called FastTree, which does a big distance matrix-based clustering. But in essence, it's a phylogenetic approach to finding OTUs that allows for reads to not overlap with each other. And you basically use a reference tree to tell you whether or not individual sequences likely come from the same part of the phylogeny by comparing them each separately to the reference tree. There's also a method that Eric Mattson developed called pPlacer that does a very similar thing. These are sort of, there are many people who have developed analogs of this sort of approach to have a reference tree, and then that allows you to find out where sequences sit relative to each other, even if they don't overlap with each other.
So what you have on the tips of this reference tree, you have the complete sequences?
It seems to work best when you have the complete sequences on the tips of the reference tree. Unfortunately, for some clades, complete sequences are not as broadly available as you would hope. So I've seen a couple of papers recently where people have been trying to generate a pseudo-complete sequence from the data itself and use that in the reference tree and then compare everything to that pseudo-complete sequence. It may be somewhat of a hybrid between different elements, but so there are attempts to use other data in addition to the full-length data that might be in a reference collection. But it is one reason to have as much full-length data as possible. For our Amphora method, we use the full genome sequence data and full-length protein coding genes to do that so that we have good sequences. I don't know how much time. But, okay, great. So I'm just going to skip over this. There's a lot of work on phylogenetic ecology, and I'd be happy to talk to people about that. I just want to point out this one other issue that I think is going to be really important, which is when you do random shotgun sequencing from an environmental sample, even when you use one of these new Illumina machines, which can generate billions of sequence reads, we still don't have deep sampling of some individual genes from particular taxa in those data sets. So the rare organisms still don't get sampled very well. And if you want to say something about the phylogeny of those rare organisms, it's going to be useful to combine data from different genes, not just to analyze each individual gene separately. And so we've been working on methods. Steve Kemble in Jessica Green's lab at University of Oregon has done a method where basically we take that concatenated alignment of all of the phylogenetic markers we used for Amphora, and then rather than placing individual genes, say RPOB and then REC-A and then something else relative to that concatenated alignment, we place all genes that have any match to any of the genes in the concatenated alignment together and use a pPlacer-like method, this reference tree method, to place individual reads relative to that reference tree. And now you can combine data from 30 or so genes, which is better than analyzing data from a single gene, to do these types of phylogenetic analyses of environmental data. So this is a tree where he's done this, and what he would like to do is basically overlay traits of some type onto the tips of the tree and then analyze this in a phylogenetic context. In this case, the the trait is what environment the sample came from. This is very analogous to a method called UniFrac, which Rob Knight and Kathy Lozapone developed to do comparison of phylogenetic structure of communities that is used a lot in the environmental world. Steve Kemble actually came from the plant phylogenetic ecology world, and there's a slightly different set of tools to do the phylogenetic comparisons that have been developed in that world. I think it's called Phylocom. I don't know if Mike knows the something from David Ackerley's lab. So it's basically to do comparisons of the phylogenetic structure of samples. So these different colors represent different samples that you have, and you can compare the phylogenetic structure and ask questions like, what is the sum total branch length in one environment versus another? How much branch length do they share? How much is unique to the particular communities? All of this phylogenetic activity is of interest to sort of phylogenetic biologists or people asking questions about diversity. It's also useful for just sorting through metagenomic data. So when we do this metagenomic sequencing, we go to environments, we grind up the sample, we have no idea which part of the sample came from which organism. We generate now these very short sequence reads from your environmental sample, and we would like to figure out, if this read came from the same organism as this read, or this read came from the same organism as that read. It's an activity called binning, basically. And there are a lot of different methods that have been developed to do this binning, sorting through the environmental reads to try and figure out what compartments they came from. Without a doubt, the best method is to have reference genomes from close relatives of the organisms from your environment. And then you can tile your sequences against those reference genomes and have a good hypothesis that if they map to the same reference genome at a very high % identity, that they likely came from at least closely related organisms in your environmental sample. And if you work on well-studied environments like the human microbiome, all the microbes that live in and on people, you're going to have reference genomes from thousands and thousands and thousands of taxa from those environments. If you work on most other environments, you're going to have nothing for a while. And so we need other methods to sort through the environmental data to do this binning. So if you don't have a reference genome, there's a lot of different algorithms that people have developed to try and sort through the data. You can look at the DNA composition. You can run an assembly of the data. You can do a variety of things. Phylogeny is another approach. So you can take the reads, build phylogenetic trees of them, and ask if they map to the same taxonomic group in the phylogenetic tree. Say this read comes from E. coli, this read comes from E. coli. You might hypothesize that they came from this read gets assigned as a nearest neighbor to E. coli, and this read gets assigned as a nearest neighbor to E. coli. You might hypothesize that they came from the same organism. And so we've used that a lot in sorting through environmental samples to do this binning. The methods that we've developed focus, again, on these individual reads, individual protein families most of the time. We actually use McGann. I'm going to come up with 17 different pronunciations. Much more than we use phylogenetic methods most of the time because it can handle massive amounts of data and give us a list of putative taxonomic groupings for sequences, and we can then take, say, all of the sequences that map to clade X and analyze them as a unit as opposed to analyzing all of the data as a unit. So two more things I want to tell you about. One is just a very, very short thing. I'm not going to talk about this a lot, which is a lot of what I used to work on and still work on a little bit is trying to predict functions for genes. When you have genome sequence data, we would like to predict functions for many of the genes in that data. And there are lots of methods that people have developed to try and annotate, predict functions for genes that are generated without experimental data, in essence. And one that I developed many years ago now is to use basically phylogenetic approaches to do this. So you take a sequence, you identify its homologs, you build a phylogenetic tree of the sequence and its homologs, you overlay functional information onto that tree, experimentally determined functions, hopefully, and then you use evolutionary character state reconstruction methods to infer the ancestral patterns of ancestral and derived character states, and you can predict the functions for unknowns based upon where they set in the tree relative to sequences that have had experimentally determined functions. And this turns out to be a powerful tool. Now, mind you, I copied this completely from Phylotyping. So this is exactly what people do when they sequence ribosomal RNAs from environmental samples. We wanted to predict the biology of this organism based upon comparison to the biology of other organisms in the phylogenetic tree, and people were even doing character state reconstruction methods. So it made sense to do this with protein function, just like you would do it with organism function. And I think it works actually quite well. There are many people who have developed this sort of general concept into high throughput approaches, and it is very powerful whatever your source of data. So you can use it for this environmental data just like you could use it for data from genome sequences. So this is an example. You build an evolutionary tree. In red are sequences from environmental data. All the other sequences are from cultured organisms, or in yellow are also environmental data. And where things sit relative to experimentally characterized genes can tell you a lot about the possible functions there. Yeah.
So isn't this technique particularly sensitive to the phylogeny you estimate? I mean, it's certainly going to be sensitive to your statements about function, but the phylogeny is also going to impact the accuracy of what you get. And if you have these short reads, then there's noise in the phylogeny. I just wonder to what extent this is going to make it difficult to actually do an accurate.
Yeah, this was done with the Sanger sequence data.
Right.
I hate it with short reads for that exact reason.
Exactly.
So we don't know what to do with the short read data because of the noise issue. Even identifying what gene family things map to, let alone the position within a gene family, has been messy for some of the very short reads. So we are not currently using this for pure Illumina short read data right now. Partly for that reason, partly because it's just so much data. We don't know exactly what to do. But this was 2 KB, 1 KB paired end sequences where, yeah.
The other question is, as you get to fuller length reads, do you think about adding things like DMDS or rate shifts to your pipeline
Yeah, I took out this slide, but yes, I mean, so I'm focusing here on just the structure of the tree itself. But there's a lot of other information in the history that includes duplication events, ratios of synonymous and non-synonymous, or conservative and non-conservative amino acid substitutions, insertion, deletion. I mean, all of those can be incorporated to really add the history. This is just adding the branching pattern part of the history, and I just punted. I mean, we don't do that as much because it's not as easy to do some of the time, but with the data we have now, it's probably easier to do. So the last thing I want to talk about is taking a step back. I generally think, as you probably can tell, that phylogenetic methods can save the world to analyzing this data. And there's a problem with everything that I've told you about, and we've noticed this problem over and over and over again, and it continues to be a problem, which is that the phylogenetic sampling of data that we are comparing to is incredibly biased. So if you looked 10 years ago now, Naomi Ward and I got a grant from the National Science Foundation to sequence the first genomes from 8 phyla of bacteria for which there were cultured species, but no genomes available at the time. And so we sequenced those genomes, we analyzed them, we thought that was, we're still writing papers on it, amazingly, and we thought that that was useful to do, and we realized after doing these first genomes from these phyla of bacteria that it was not enough. And when I moved from this place, Tiger, to UC Davis, had an adjunct, I still have an adjunct appointment at the DOE Joint Genome Institute, and I somehow convinced them to do a bigger scale version of this. And now what we've done is basically gone through the ribosomal RNA tree of bacteria and archaea, identified the branches that have no representative genomes available, but a cultured organism available, and sequenced a representative from that branch. And we've done about 350 genomes now to fill in the tree of bacteria and archaea. The data is released to GenBank with no restrictions on its use as soon as we get assemblies. So anybody here can analyze all of that data. And we've been analyzing that data to try and figure out what the rules are that have come out of this. And I'm just going to quickly tell you some of the things that we found. So for example, this didn't necessarily have to be the case, but as we walked through the ribosomal RNA tree and then sequenced genomes, that was a pretty decent way to find lineages for which the average genome phylogeny also looks to be novel. So it was possible that the ribosomal RNA novelty would be completely useless. I mean, it didn't seem likely, but could be completely useless to finding organisms that on average their whole genome seemed to be somewhat novel. And that was not the case. That is, there weren't any organisms that seemed to be really novel from their ribosomal RNA point of view, and yet when we looked at their genome, were really closely related to something else that we had genome data from. Now, this doesn't mean the topology in the ribosomal RNA tree is perfect. There are many cases where the topology of either the average genome tree or hundreds of individual gene trees is different from the topology in the 16S ribosomal RNA tree. But the general sort of novelty in the 16S tree seemed to be a good predictor of general novelty in the average sort of genome phylogeny of organisms. The main reason DOE agreed to let us do this project was the argument that we basically took from people like David Hausler and others who had argued this for vertebrates and for plants, that is, that phylogenetic novelty should help us in annotation of genomes. That is, by having a good sampling of genomes from across the tree, that should help us do things like annotate even E. coli. And I'm not going to go into all the details here, but we basically showed in every type of annotation method that we looked at, phylogenetic diversity helped us in annotating various aspects of genomes. Probably much more important, at least from an evolutionary point of view, is this last issue here, or second to last issue here, which is this was sort of a really big roadblock to doing this project. We argued that it was likely that phylogenetically novel organisms in the ribosomal RNA tree would have biological diversity, more biological diversity than randomly closely related organisms would have. And the basis for this was, sort of straightforward looking at phylogenetic trees and what I talked about before, that some novelty should have evolved in the branches that represented these unique lineages. It didn't mean that you couldn't have novelty evolving among closely related organisms, but on average, we expected that there would be some level of novelty, higher level of novelty in these organisms that were distantly related from other taxa. And the reason this might not be the case in bacteria and archaea was mostly due to the possibility of lateral gene transfer wiping out the signal of the core phylogeny of these organisms. That is, the ribosomal RNA tree or the REC-A tree or whatever, if those were phylogenetically novel, but the organisms were swapping their functional genes sort of at a very high rate, then a phylogenetically novel organism would not actually be expected to have any particular higher level of functional or protein family diversity than anything else. To test this, we did a bunch of different things. This is just one example of the way we tested this. We basically did a rarefaction curve for protein family diversity for different sets of genomes that had different level of relatedness. So, and we did this in a couple of ways. I'm going to show you this one way that we did it here, which was we took all the genomes, we compared their proteins to each other, built gene families from those genomes, and then for different genome sets, we asked, as we add a new genome to that set, how many new protein families does it add when we add genome 2, genome 3, genome 4, genome 5? So we did this within a species. These are all, I don't know if people in the back can see it, but it's a not very high slope here for within the species of Streptococcus agalactiae, which was a species that people had proposed the existence of something called the pan-genome. So it turns out every genome that you sequence in this group gives you new sequence. I mean, it's every, new strains of Streptococcus agalactiae, each one has new protein families. And we found the same thing, but the rate of recovery of protein families is pretty low, 121 about per genome that was sequenced. If you do this for organisms that are more distantly related on the ribosomal RNA tree, the Enterobacteriaceae, this is one family, you see a higher rate of recovery of protein families per genome. If you do this for a phylum, the Actinobacteria, you see an even higher rate of recovery of protein families per genome. And if you do this for our Genomic Encyclopedia project, you see sort of the highest rate of recovery of protein families per genome. Now, this isn't particularly surprising, but it basically means that in terms of protein family space, there is some signal there in the phylogenetic diversity of these organisms. That is, that they are more likely to have more protein families between them if they are more distant on the tree. Another way of saying this is that synapomorphies exist in bacteria and archaea. That is, shared derived traits do in fact exist in clades of bacteria and archaea. Not saying lateral gene transfer does not happen, not saying it's not important, just saying that it has not wiped out all signal of vertical evolution in these lineages. And that means that if you want to discover protein family diversity, an important vector in it is phylogenetic novelty of organisms. It's not the only vector, but it's an important vector. I'll skip over. We're trying to figure out which lineage has the highest rate of recovery of protein families, because it's not uniform across all lineages, because those might make more sense to sequence more deeply than other lineages. And then I'll just end, in essence, on this, our dream, which was my real goal in sequencing genomes from across the tree of life was to improve all of these analyses that I've told you about for environmental data. So if we want to build phylogenetic trees of RecA, we need reference data for RecA. If we want to build phylogenetic trees for RPOB, we need reference data for RPOB. If we want to do anything for Mugen or Megan or for any method that we want to take advantage of comparative data or phylogenetic data, we need to have genomes sampled from phylogenetic diversity. So we went and redid many of these analyses that we had done looking at phylogeny of environmental data with these new genomes. Things like the phylotyping, which can't be done without a good sampling of genomes. The binning can't be done without sampling of genomes. And this genomic encyclopedia project improved this, but barely detectably. I mean, just the most trivial level of improvement you could imagine to these environmental analyses. And I'm just going to skip over a couple of other things to show you why. The reason why is because we have not even remotely scratched the surface of PD, of the phylogenetic diversity of bacteria and archaea. If we take the ribosomal RNA tree of life and plot PD, which is the branch length in the tree, and we measure that PD with an algorithm by Mike Steele. I don't know if he's here. He's coming in a couple. Oh, he is here. So we use his algorithm for doing this, and we measure PD in the ribosomal RNA tree, and we sort it by the most PD. You see that some organisms in the tree are distantly related, of course, to anything else in the tree. Some are close. And we did this first for organisms for which genomes were sequenced before we started this project. So some of the genomes were pretty novel compared to other genomes that were sequenced. Some were basically the same, that is all the anthrax genomes, all the E. coli genomes, etc. Our genomic encyclopedia project, each genome added a lot of PD. It better have, that's how we picked them. So if that wasn't the case, we were in deep trouble. If you look at the cultured described organisms of bacteria and archaea, of which there are some 7,000, we would only need about 1,000 more genomes to get half of the total PD in the described organisms of bacteria and archaea. We're going to get there with this project. So we will, some of them are very hard to grow, but we will eventually get to half of the total PD of the cultured organisms of bacteria and archaea. But if you look at all the ribosomal RNA data from environmental samples, and we did this only with full-length data from 4 years ago, we would need 10,000 genomes to get to half of the PD from all of the ribosomal RNA data that was available in that full-length data at the time. I would estimate now that we would, based upon analyses of some of the new data, we would need about 200,000 genomes to get to half of the currently known PD of bacteria and archaea. So, pardon my French here, no shit that our method, that our sequencing 56 genomes did not help in sorting through all of the environmental sequence data on the planet. Now, this is hard to do. Sequencing genomes from uncultured organisms is hard, but there are projects underway to do this. So the Joint Genome Institute has now spun off a project from our genomic encyclopedia that's called the Uncultured Encyclopedia. Tanya Woike there is running this project, and they are going through the branches where all the PD really is now and trying to figure out ways to sequence the genomes of those organisms, either by whole genome amplification or finding enrichment cultures where some of those organisms are abundant, or environmental samples where they're abundant, and then we will fill in the whole tree and be able to sort through all the environmental data. And I'll just mention one other thing. Most of the methods that we use to analyze sequence data do not explicitly take advantage of having broad phylogenetic diversity of samples. So many of the methods that people use could be re-engineered to better take advantage of this new sort of genomic encyclopedia data. I had some slides on it, but I won't drag this on and on and on forever. And so I'll just leave it there. I think that phylogeny is an important tool. Obviously, I think that. but it's not just a tool in analyzing data, it's a tool in helping guide us in terms of what things to do. Now, plant and animal people would say, duh, that's what everybody is doing. And for whatever reason, the bacterial and archaeal people had not been doing this. I think largely because of this fear of lateral gene transfer swamping out all signal of phylogeny. But it clearly does, in fact, help to sample through the tree. So I'll leave it there.
I think we've got about 2 minutes until lunch break.
Yeah.
So
Yeah.
So, with respect to your conclusion that there is a signal despite all the lateral gene transfer,
Yeah.
so how would you really suggest to reconstruct that tree of the signal? I mean, because again,
Well,
gene,
we're using methods from the previous speaker to try and do this. So we're actually using Bucky to try and do, in essence, whole genome phylogeny for bacteria and archaea. You've probably heard from people in my lab about this. I don't know, Aaron Darling. And, so that's one approach that we're trying to use. I think that in the end, what the answer really is that there's sort of a hybrid between short-term vertical evolution, in many cases, and short and long-distance lateral gene transfer events. And then you want to incorporate everything. I mean, like David Hauser said, we want to have a reconstruction that incorporates duplication, deletion, gene transfer, rearrangement, and vertical evolution. And that's what, I mean, I'm not doing this, but there are people who are working on models, Bayesian and likelihood-based models, to try and do that with bacterial and archaeal data. And I think that there is signal there. It's just that the extent of the reticulation is much greater than we see in many plants and animals, and it's much more complex. And so reconstructing it is tricky, especially when we don't have good sampling. So for example, something shows up in a genome, it seems to be laterally transferred. We never have a donor, basically, because we don't have a good sampling of data to try and figure out. We can say something is anomalous, but it's very hard to say where it might have come from right now. So that part of the models is pretty bad currently. But I think it's solvable by these integrated sort of Bayesian or likelihood-based models that incorporate everything.
Yeah. Question, to what extent are people using the existing number of bacterial genomes as sort of known quantities? This issue of metagenomic diversity and short-read technology, it's just going to get worse in some ways. But if you're interested in knowing how well things can be binned and clustered and assembled under those conditions, surely you can take all the known sequence genomes and you know them because they're sequenced and you can just shred them up into pieces and then play around with the process of putting them back together and see how well it reconstructs true history.
Yeah, so you can definitely do these simulations. One problem with these simulations is that so we've even done in vitro simulations like this. We've grown up organisms. We published a paper on it last year. Grew up 10 different taxa. I counted cells, combined together the exact same number of cells of them, and then ran them through metagenomic sequencing. And that is useful to do, but in the end, they're not artificial communities. They're very artificial, especially in terms of the population genetic structure within some of those groups. So until someone runs some more pop-gen simulations on the data, which I have heard people talking about, taking a genome and then simulating a population from it, and then putting that into the metagenomic simulations, until people do that, I'm not very confident that the simulations that have been done are that useful in testing the methods.
Probably the last question.
I notice you end with something that looks a lot like a starfighter, and it just makes me wonder to what extent your saga of undiscovered phylogenetic diversity is really a saga of undiscovered replicates from this giant star population. And if that's true, the story is less about phylogeny than it is just about sort of
Noise.
Undiscovered replicates.
Yeah, so the ribosome RNA tree frequently collapses into a really nasty star phylogeny, but when we do, I mean, if you believe some of the whole genome level phylogenies that we're doing either with concatenating alignments or with analyzing each gene separately, there is superstructure there for many of these lineages. So many of the phyla are coming together into supergroups that are very consistent across all the genomes of those lineages. They're still, it's still star-like in many ways because they're deep, they're short internodes leading to these supergroups, basically. So it may be that a lot of it is what you're saying. And I'm also worried about things like amino acid content convergence and GC content convergence in different lineages. If there was a star phylogeny, we'll group them together artificially based upon whatever converged at some metric like that. So I think there's a combination of both. There is some superstructure. We're trying to find out what those supergroups are, and I'm ignoring the bottom. I don't know what to do with the bottom. But there is superstructure there that we are finding now that we get genomes for many of these lineages. Does that sort of answer the...
If it's very short, we should really get that.
I was just wondering what the status is of sequencing uncultured genomes. I know there was a single cell genome. How many people are actually using this right now? How much data is generated?
Well, there are multiple groups that are doing this. So the Joint Genome Institute group has generated about 100 genomes now from uncultured organisms. Now, the genome quality varies enormously because the amplification step usually with this MDA, multiple displacement amplification process, is pretty awful. So you amplify from a single cell, you don't get either exactly perfect data out of that or complete data out of that. So they're working on methods to either combine together separate amplifications from closely related taxa, or what I think is going to be in the long run is to do enrichment cultures and to do environmental samples that have a highly abundant representative of these groups, because then you're not dependent upon these amplification methods that seem to create almost more problems than they solve. But there's a big human microbiome project that has done a few hundred genomes as well that are by single-cell genomics. There was one paper they just published on this a couple of weeks ago, I think, or a month ago, with the first 8 or so of these. So they're coming. Some of them look pretty good, but 80% of them are awful, and it's really expensive. So we're not doing it myself, let's put it that way.
Okay, well, thank you once again. We will resume at 2:30.